All right, last time where we stopped, we were talking about ultrametric trees and ultrametric distance. Okay, and so let me just remind you that we have a symmetric matrix. I haven't written the bottom portion because it's just the flip of the top. And if you can create a tree where each row labels exactly one leaf, and you have one number at each node, and walking from the top down, those numbers are strictly decrease. And for any pair of rows, let's say A and D, when you walk up to the nearest ancestor, of both of them, nearest common ancestor, the number that's written there is the AD entry in the matrix. Then this is called an ultrametric tree for this matrix. And the point is that this looks like an evolutionary tree if we think of these numbers as somehow time in the past, or something that's proportional to time in the past. Okay? This may not really be 8 units, but from that number 8, you can convert to however much time or how many generations or whatever your unit is in the past. But the data, so this is what we want to build. If you have species or organisms A through E, and you want to understand their evolutionary history, this is the output that you finally want. The kind of data you have to work with comes from sequences. This is a sequence-oriented course. Of course, in the past, there were other things that you could work with, morphological features. But let's concentrate on sequence because it's a little bit easier to think about. You have sequence data from these different species, and you can do comparisons of those sequences. Let's say pairwise comparisons, and you find that the distance between A and B is 8 under some objective function. Now, this is a minimization objective function, like a distance. So we're trying to minimize the number of mismatches or spaces. The reason I know that is because further back in time is a larger number. You expect that things that diverge just a little bit in the past should be more similar than things that diverged quite a bit in the past. So you get bigger numbers as you go further into the past. So this is some kind of distance. And smaller equals closer. Okay, closer in time anyway, since divergence. So this is the kind of data that you can collect by looking at the sequence. And this is the kind of tree that you want that explains the history of these organisms. And what I want to try to explain today is how you recognize whether or not your matrix has an ultrametric tree. Not all matrices do. Can somebody off the top of their head see a condition that says that a particular matrix would not have an ultrametric tree? Is what? Is what? Well, okay, now the reason why it may not have one in terms of biology, convergent evolution and other things may be true, but I just mean just in terms of numbers in a matrix. Is there some simple thing you can look at and say, if it has that property, then you're cooked? There's no way you can have an ultrametric tree. Yeah? Say it again?
Yeah. Say if you have the matrix A, B, and C.
Yeah.
And then A and B and A and C both were the same number.
Well, A and B and A and C being the same number doesn't sound too bad. A and B, I see. And A and C, if they were the same number, then you would expect them all to meet at one place. And we said that these trees have to only have 2 children. Yeah, that would be certainly a condition that would be bad. Right. Another one, if you have 1, 2, 3, 4, 5 leaves, it turns out no matter what the shape of the tree is, as long as every node has only 2 children, you always have only 4 other nodes in the tree. No matter what tree I draw, with 5 leaves, as long as each interior node only has 2 children, it's easy to argue that you have to have exactly 4 other nodes in the tree. Okay? So if I have more than 4 different kinds of, four different numbers in here, okay, off the diagonal, if I have more than 4 distinct numbers in here, then I'm in trouble too. That's another condition. So we have the 8s, that's one type of number. We have the 3s, we have 5s, and that's only 3 distinct numbers, but that's less than 4, less than or equal to 4. So we're not in trouble. We don't know necessarily there's a ultrametric tree. Okay, anyway, so my point is that most matrices of numbers you make up is not going to have an ultrametric tree. This is a very special situation when that occurs. Okay? But, let me put a theorem here. A matrix M has an ultrametric tree if and only if for any 3 rows ijk, this thing was a distance matrix D, okay? The max of Dij, Dik, and Djk is not unique. That's okay. A lot of words again. Let me try to illustrate what that means. Okay. Well, let's just take 3 rows. Okay. Somebody in the audience that I've never met before, suggests 3 rows in this little example. This is an easy question. ABC. Okay, very good. ABC. And if I look at DAB, DBC, and DAC, what do I have over here? DAB is 8, DAC is 8, and DBC is 3. So 8, 3, 8. And so what you see, that the largest of those, the max of those, which is 8, is not unique. There are 2 of them. Okay, there's always a tie for the max. So that's another way of saying this not unique. There is a tie. A tie for the max. Now, one side of this theorem is very easy to see. If this matrix M is ultrametric, okay, then this certainly has to hold. How do we argue that? Well, if it's ultrametric, then we know by the definition of what it means to be ultrametric is that there's some tree like that represents the matrix. And if we just look at the 3 leaves that are being enumerated, well, I'll do this abstractly. IJK are 3 leaves that are being enumerated. And we walk up from those leaves to see what portion of the tree is being described by them. And as you walk up until you see a common ancestor of some 2 of them, and then finally you walk up till you see the common ancestor of 3 of them. What does it look like? It looks like that, or actually there are 3 possibilities depending on which 2 of these meet first. Okay, but they all have the same form. I could change around the labels and we would have all the possibilities. Okay, so you have one pair of this that's meeting first. And because the numbers are decreasing, this number here, well, we said it was supposed to be Dij. If the tree is ultrametric, this has to be Dij. And what's the number up here is Dik, and that's equal to Djk. All right? So if the matrix has an ultrametric tree, then whenever you look at 3 rows or 3 leaves, it has to have this property that the max of these 3 numbers is not unique. It's a tie. The other point was that Dik, these numbers are bigger than that number. So these are the max, and there are 2 of them. Okay, there's a tie there. So that's one direction of this theorem. The other direction is that if this is true of each triple, then you can actually form an ultrametric tree. Okay? Now, I'm not going to prove that side, although I have notes that I'll put on the web, I haven't done that yet, that do have a proof of that. And in the biological circles, this condition here is called the relative rate test. So in the evolutionary world, building evolutionary trees, this condition is referred to as relative rate test. But it's just this standard ultrametric condition. Okay, well that tells you an algorithm, by the way, for determining whether or not your matrix has an ultrametric tree. You just enumerate all triples. If you're writing a program, you'd have 3 loops, and you'd pick I over all the ranges, and J over all the ranges, and K over all the ranges. And then you'd look up the D values for those pairs and check them to see whether this condition held for each triple. That wouldn't actually build the tree for you, but I'll show you in a minute how we can build the tree. All right. What's next is the most important theorem here, which is that if M, that's the matrix, has an ultrametric tree, then it is unique. There's only one tree. We have this matrix the matrix gives you a tree in only one possible way. Okay? So why is that really important? If this is data that comes from sequence comparison, and we think that the differences, the distances between 2 sequences are somehow proportional to the time since those organisms diverged, if we really think that the sequence difference is telling you something about the time since divergence at some proportionality rate, some way of converting this number 8 into some actual distance in time. If we think that the numbers have that property and they're ultrametric, then it says that this is actually the historically correct tree. It isn't just some nice representation of this matrix, and maybe there are many, many other representations, many, many other trees that look different but have the same property. What this theorem of uniqueness is saying is that if this is ultrametric, and moreover, if we really believe that these numbers are telling you something about time, these numbers are proportional to the actual time since these organisms diverged, then you can completely and correctly infer what the history of that divergence was. There's only one possible explanation for these numbers if they're ultrametric. It's this tree. Okay? So that's very, very powerful theorem. Very powerful. It says that ultrametric data is likely to be biologically significant. Now, another way of arguing this is that most matrices that you would invent are not ultrametric. In fact, not even close. Get lots of violations of this by a lot. So if you have data that really is, if it either really is exactly ultrametric or it's very close to being ultrametric, it really is suggestive that A, this data really, the numbers that you're working with really are proportional to time since divergence, and B, that the tree you get out is the historically correct reflection of their evolution. Okay? So ultrametric data is very, very significant if you find it. One of the things that gave this whole area a boost was a large project that was done in the 1970s that compared distance between sequence and birds. This was, I don't know how many hundreds or thousands of bird species were done. They're compiled in an enormous book. They didn't do it by looking at sequence. They did actual laboratory hybridizations. Anyway, they had some laboratory method for calculating the similarity of the molecules from different birds. And they had, from those, they came out with a particular number for each pair. Now, there's a lot of controversy, actually, about whether they did their chemistry right or their biology right. But the point is that they had maybe 1,000 different birds, and the data was almost perfectly ultrametric. So when it's so rare to have ultrametric data, the only reasonable explanation, well, on the one hand, it's rare to have ultrametric data. On the other hand, if the data really does reflect evolutionary time, then you expect it to be ultrametric. So the only explanation, really, for why you had ultrametric data was that it was really reflecting evolution, and therefore the tree was the appropriate reflection of the evolutionary history. Of course, there's always a third explanation for why you have ultrametric data. Why is that? Some people are laughing, so they know the answer. I mean, in real science, the third explanation is that you fudge the data somehow, either consciously or unconsciously. You wanted it to be ultrametric, so kind of made it ultrametric. After many, many, many years of sifting through lots of different kind of data, and you look at it, and this one doesn't quite work out. Well, it must have been because I didn't turn the knobs quite right. We'll throw that out. And then this experiment I did on Tuesday looks better. That must be right, so we'll keep that one. Who knows? There's a lot of controversy about this. But the conceptual point is this, again, if you see data that seems ultrametric, the only explanation anybody's ever proposed is that, yeah, that's really, those numbers are proportional to time since divergence, and therefore the tree you get out really does capture the evolutionary history. Okay. Now, another thing that happened at the same time the bird study was done was which really, again, helps to explain why ultrametric data is so suggestive of evolution, is the molecular clock theory. Thesis. Has anybody seen this before? What is the molecular clock thesis? Nobody's seen this at all? This is one of the great, well, this goes back to the first lecture, in terms of the dogmas that we talked about, or the simplifications that we talked about, for a while this was one of the great accepted simplifications. Now it's in a great deal more trouble. But this basically said that absent of selection, mutations that were occurring in molecules were happening at a uniform rate. Okay, and this could differ by different protein, but the point is that if you looked at a particular protein in birds or crocodiles or humans or all the different organisms, and you were able to see how that molecule was mutating over time, and you were not looking in the functionally important parts, but sort of the parts that were not under selection, you would see mutations happening at a fairly constant and uniform rate. So this was that mutations outside of functional domains occur at a constant regular rate. Okay? So there's a clock, all right? So that if you took a look at two sequences, and by again, this is for a particular protein. Different proteins have different clocks. That was certainly known. Some proteins are evolving at a much faster rate than others. But if you focus on a particular protein and you look at its rate of change, if you could look at its rate of change over a number of different organisms, then the belief was that these changes were occurring at a more or less constant rate. Now, by the way, more or less, you know this has to do with long enough time periods. It's not like there really is a clock, and somewhere in the molecule it says, well, it's been 23 hours since the last change. Now I'm going to change again. So this is something it's statistical. You're seeing this over a long enough range of time, and you shouldn't be surprised if you see that this isn't exactly right, even if you could look in the past, which we can't. But even if you could absolutely see what was happening in the past, it shouldn't be amazing that there's some minor deviations. But statistically, over long enough time periods, this was the belief. Okay, I'll connect this to ultrametric in a minute, but just to make this point that we're only talking about kind of statistical generalizations over long enough time, it reminds me of the joke that I'm supposed to tell at this point, which was at this point in the lecture, the professor is supposed to ask, how long has it been since birds and crocodiles or birds and giraffes diverged? By the way, does anybody have any idea? You're all biologists out there. 40 million years? I don't know. I'm making that up. I have no idea. Let's say 40 million years is sort of the canonical number. But the point is that the professor asked that question, and one of the students said, 40 million in 1 year. It's a very exact answer. And the professor said, well, how can you be so exact, 40 million in 1 year? And the student said, well, you see, the thing is, I took this class last year and I failed it, but last year you said it was 40 million. Anyway, so this is just rough. This is very rough. All right. But how does this help? How does this help? How does the molecular clock thesis help strengthen the belief that ultrametric tells you something about evolution? Because these numbers have to do with differences between the two molecules. Why should they tell you anything about time? Remember, we said these numbers, if they're ultrametric, will give you a nice tree, and that tree will be meaningful if we believe that these numbers are proportional to time since divergence. These numbers will be meaningful to you as biologists if these numbers are proportional to the time since the two sequences the two organisms that sequences were taken from, diverged. Now, why should that be? Why should somehow the difference, some measure of difference between two molecules have anything to do with time? Well, it's the molecular clock hypothesis. Let's just, for simplicity, say that we started out when two molecules here, we're looking at a particular sequence in some bird, and here's giraffe. And at their ancestry, at their common ancestry, that sequence was the same. That's what it means for it to be a common ancestor. Okay? And now, since divergence, this one started changing. It got some mutations in it. Okay? And this one changed. So it has some mutations in it too. Now, it's possible that two mutations actually happened at the same location. But for simplicity first, if those mutations don't happen at the same locations, then when I do this alignment, I'm roughly going to see, you'll see identity. When you do a good alignment, you'll see identity on these sections, all right? Where there weren't any mutations, you'll still see the identity. But you'll see a number of differences, mismatches, let's just say, without insertions and deletions, which complicate things a little bit. But let's just say the mismatches, the total number of mismatches is equal to the total number of mutations that happen along here and happen along there. But if the molecular clock is to be believed, the number along this side is roughly equal to the number along that side. So that when you compare the two sequences and you find that there are, let's say in this case, 6 differences, then that tells you that the time since divergence is however much time it takes to have 3. That's 6 over 2. 3 mutations, okay, in a single sequence. So that's why if I take the comparison here, 6 mismatches, I can then say, oh, I think the time since they diverged was however much time the molecular clock needs to have 3 mutations in that particular protein. And therefore, you can go from a number like 6, which you get by comparing the two sequences, to a number which is proportional to the actual time since these two sequences diverged. And you don't need to know what that rate of proportionality is in order to build the tree. You don't have to know, how long it is in that protein between mutations. In fact, people figured that out by thinking backwards. If they happen to know from fossils what this point in time was, or from some other reasoning what they think this point in time is, then they look at the molecules and they say, oh, there are 15 changes. Okay, or let's make it an even number. 15, okay, so they look and they see 30 differences between the two molecules, so that's 15 time units. And they know what this absolute time is, 40 million and 1 year ago. And then they can divide that by 15 and say, okay, the clock ticks at the following rate. Every however many years or how many generations, that's what we think a single mutation is. Okay. Now, all of this is complicated by the fact that you can have mutations at the same place. But under a certain probabilistic model, you can figure out how many coincident mutations you should expect. And therefore, it's easy to work backwards to say, if I really observe 6 mismatches, whatever, it was really because of, by the way, more mutations or fewer mutations? If you observe a total of 6 mismatches. That's what you would get if all the mutations happened at different locations. If they happen at the same location, so let's say we had an A here, this mutates now to a C. At the same location, this mutates to a T. When I look at these two, I count that as one mismatch. I don't realize it was actually 2 mutations. Okay? So when you do your comparison here, you can undercount the total number of mutations. Okay? You don't overcount the number of mutations. You can also have back mutations, for example. You might have this A turning into a T at some point in time, and then sometime it turns back. Okay? So again, that undercounts the number of mutations. But the point is that that's been studied, and so you can have a formula which tells you if we have 6 observed differences, we really think it was 8. And then instead of 6 divided by 3, we take 8 divided by 3, and we think it's 4 time units since divergence. But anyway, that's all details. The main concept, the main point is that because of the molecular clock thesis, it's believed, therefore, that you can just take a look at 2 sequences, look at some measure of their difference, and think that that's somehow proportional to the time since they diverged, or these numbers corrected by the fact that you may have undercounted somewhat. But once you correct for undercounts, then the numbers should be proportional to time since divergence. And if the numbers are proportional to time since divergence, and this is ultrametric, then you really feel confident that you have the tree in hand. So this is the tree of truth. This is the true tree that you're looking at, and you've got it. And as I said, in the early 70s, people were finding ultrametric data all over. Wherever they looked, it all looked ultrametric. Now things don't look so ultrametric, and there's sort of a questioning of why people were so convinced early on. But still, the basic game plan is very important. Okay, now how do you actually build a tree from the ultrametric data? And it really turns out that almost anything you do that's sensible works. When the tree is ultrametric, when the data is ultrametric, it's somehow screaming out for an ultrametric tree. Almost any idea you come up with will work to build it. I'm sure you could put your mind to it and find some that don't. But if you just think again, these numbers are telling you time since divergence, okay? So small numbers mean that the two leaves, the 2 organisms, they're probably neighbors in the tree. So you just take the smallest number, could be ties, but that's AE. You put AE together, there's a 3. Next smallest number is BC. Well, it's a tie, okay. B and C are not related to this, so here's B, here's C. Put those guys together. Next smallest is 5. That's, what is this? A, B, C, D. So that's DE. Now, E is already in the tree, and D is not. So you represent that as this way. Remember, we're trying to build an ultrametric tree. 5 is bigger than 3, so it has to be above it. So D and E, but they have to have as their ancestor that 5, so it forces that kind of connection. There's another 5 in here, that's AD. But what? I just did AD. DE. Well, okay, no, I did DE. DE, and now we're learning AD, that's just reinforcing that. There's nothing more that's happening there. And then you have an 8, that's C and D. And put those together because C is in here and D is in there, so they put that together, and there's your 8. Okay? And I could have worked this algorithm backwards by throwing away big numbers. There are zillions of ideas that people have come up with which always come back to the same tree. Well, because the theorem says the tree is unique. Okay? And just about any algorithm you dream up that's somehow sensible is going to give you that unique tree. Okay? So that's the ideal case of ultrametric data. Now, in reality, most data is not ultrametric, as I mentioned. And I hope next time I'll have time to tell you about the algorithm people use when data really isn't ultrametric. But there's another compromise before the real world, which is additive data. So sometimes the data is not ultrametric, but it is additive. And what additive means is that I can draw a tree. I start with a matrix, again, as this we did here. And I have a tree where I put numbers. So some number, 8, 3, 2, 1, 4. The numbers now are not on the nodes, they're on edges. But again, they're going to represent time. The time it took evolution to go from here to there, the time it took to go from here to here, and so on. And for two organisms, if we add the numbers along the path between them, if that number is the same number in the matrix, that's true, and that's true for every pair, then this tree is called additive. And additive is also, that's a property you absolutely expect from an evolutionary tree. If you think about it, every ultrametric tree is additive. But not every additive tree is ultrametric. Okay? So we'll talk about this next time. The reason I'm stopping a little bit early is I have to pass out the evaluations, which really are quite important in the scheme of the university. And I would like you to take the time that we have left to actually fill these out. I also need somebody to deliver them to the department because they don't trust faculty. We could cheat. So when you have yours done, come up and put it in this envelope, and somebody please come and volunteer to bring it to the department. I'll be here to answer questions for the next few minutes. There are two, by the way, there are two forms. One is for me and one is for Sasha. And I should mention that Sasha put together the lab this morning that you all did, which I think was a very nice lab on GenScan. That was her project.